There's so many things. It's too many good ones. I want to start over. My name is Emily Bulger, and I'm a PhD candidate in Benoit Bruneau's lab at Gladstone. When I started doing my rotations and kind of, testing out different labs, something that really stood out to me about Gladstone was how much community was emphasized kind of at every level. So both at the administrative levels with support systems like Gladstone Go, our graduate organization, and also just within the labs. And some of the best advice I got when I was looking for a lab was to try to pick a place that was filled with scientists that I wanted to kind of grow up to become like. I think that that's really manifested in my time here. I love how collaborative it is. I think that the resources we have access to are pretty incredible, especially like some of the microscopes and just like the ability of just the precision that we can get with science and the tools that are accessible, I think is really incredible. And also just in talking to all of the talented scientists around us, I feel like every conversation I come away from just really getting a better understanding of my project and thinking about it kind of with a new lens. And I really appreciate that diversity of ideas. Definitely interested in science as a child. I don't think I always knew it was what I wanted to be when I grew up, but from a young age, I think that my family really supported curiosity and exploration. My grandpa was a curator for the New England Aquarium and also a little bit for the Cal Academy. And so I guess, the science gene is kind of in the blood a little bit. Kind of big picture goal of my research project is understanding how the dosages of different genes influence how an organism develops. And so I'm specifically looking at a gene called brachyury, which in Latin roughly translates to no tail. And that's because mice that don't have this gene are missing pretty much their whole posterior trunk. And mice that only have one copy of this gene are missing their tail and tend to have kind of malformed structures in their pelvis and lower vertebrae. And so what that translates into for humans tends to be malformations in spinal cord development. And so what I'm doing is I'm taking human stem cells and I have stem cells that have 0 copies, 1 copy, or 2 copies, which is normal, of the brachyury gene. And I'm growing them in a dish in a system that kind of mimics early development. And so my cells look a bit like a bullseye. It's a round colony of cells, and they have like pre-neural cells in the center, pre-muscle-like cells kind of in the next ring of the bullseye, pre-endoderm, which is like gut cells kind of in the next ring, and then embryonic tissue, pre-embryonic tissue kind of around the outermost ring. And so when I put my cells with my different copies of this gene brachyury into this platform, I can then assess kind of how expression of this gene influences what type of cells these stem cells can turn into, and also kind of how they interact and communicate with one another. With the overall goal being trying to understand sort of how this gene regulates the cells that are important in the early embryo, kind of in the first 2 weeks after fertilization, and how that goes on to build out the body plan and eventually the spinal cord and the adjacent muscles around it. I mean, I've had a lot of influences that kind of helped me go toward the science route. I think my family had a huge role in that, and just instilling curiosity and wondering kind of how things worked around me. I've also been fortunate to have a lot of really good role models, both through undergrad and through research associate positions later on, and then now through grad school, who have kind of reinforced just that curiosity element and kind of not letting that get in the way of, whatever the next grant or publication or, whatever kind of formality of the process is, just making sure curiosity is at the heart of it. If I could understand and be able to communicate in any language I wanted at any time, that would be pretty incredible. Just, getting to see the world in different ways and communicate and see how other people view the world. And traveling would be a plus. Jane Goodall, mainly just because of how big of a pioneer she was when I feel like women didn't necessarily have a very prominent role in the sciences, and she really opened the door in a lot of ways, especially in their science communication skills, to getting the public engaged, with the process of science, making it much more normalized for women to be a big component of that. And, the ecologist in me just thinks that the work that she's done is really fascinating, and I think it would be an interesting conversation.